AP Biology Midterm Review. Disclaimer, this review is only intended to be a review of Unit 3 concepts and not a replacement for in-class content. Some information is simplified, and studying is, of course, still important to be successful. We won't also not cover photosynthesis in depth, depending on time. Good luck. AP Biology's big idea number 2, energetics. Biological systems use energy and building blocks to grow, reproduce, and maintain dynamic homeostasis. That word homeostasis means to maintain an environment inside of cells that is unchanging. Okay, in general, enzymes speed up metabolic reactions by lowering energy barriers. So it takes less energy than it normally would for a reaction to go from reactants to products. And that's what enzymes do. So even though a chemical reaction in the cell may be spontaneous, meaning it requires no outside energy source to react, it can be so slow that the organism would die because it doesn't have time to receive the energy from the reactions. For instance, if you put sucrose in water and dissolved it, over time we would not expect this reaction to happen at all. It would not combine with water to form glucose and fructose. However, there is an enzyme that does this called sucrase that hydrolyzes sucrose, breaks it into the monomers glucose and fructose. Okay, and so the solution is enzymes. Enzymes are proteins that are catalysts, and catalysts speed up chemical reactions. They are also not consumed, which is a big deal, so we can recycle them in the cell. How enzymes speed up reactions. In general, it takes an input of energy, sort of like a spark to start a fire, to get a reaction going. And enzymes speed up these reactions by lowering the energy that it takes, the activation energy. So you can think of it as the enzymes make them have less effort to go from reactants to products. Okay, and so we notice this energy diagram. So energy goes up this way. Notice this is an exergonic reaction because the reactants are on the top and products are more stable on the bottom. So it wants to become more stable. All right, and then let's look at the activation energy without an enzyme. That's this peak right here. So that's the normal peak. Okay, and then we have, when we introduce an enzyme, notice it catalyzes by lowering the activation energy barrier, going from reactants to products. So notice there's a smaller hill there, activation energy. Notice delta G is unchanged. Okay, we do not change the free energy or the equilibrium, meaning the placement of the reactants and products concentrations. The whole point is enzymes only speed up reactions that would eventually happen anyway. Enzyme's shape or the structure determines what reaction it catalyzes or the function. So the point is, just like in many things in biology, the structure determines the function. So the structure of the enzyme determines what reaction it catalyzes. And this allows the cell to control specific reactions and processes. We can't just have a million different reactions going on in the cell at one time. That would be chaos and lead to death. So a good analogy is, think about a kitchen. There's all kinds of specific tools for each job. You wouldn't use a spoon to cut a piece of bread. And so enzymes are specific for their substrate that they react. So they only use a specific substrate, and the substrate is the thing that an enzyme is reacting, and it happens in a place called the active site of the enzyme. So here's the enzyme right here. Notice that the active site is sort of a divot in there. And this is called the induced fit model. This is where the substrate binds to the enzyme. And notice the enzyme sort of wraps around. So if we have the imagine this is the enzyme, this is the substrate. So notice that it's not just a direct fit like a key. Turns out the substrate comes in and the enzyme sort of wraps itself around, making a tighter seal. Okay, so the substrate is right there in the active site. The enzyme and substrate form a complex, and it forms around it. And that's called the induced fit model. And the substrate is held in the enzyme-substrate complex by weak bonds, like hydrogen bonds. Okay, and remember, it is a protein, so it does have a 3D shape, and that allows it to be specific in what it reacts with. Okay, so in general, we have substrates enter the enzyme. Then they form around the substrates. This lowers the activation energy of the reaction, speeds it up, and it causes the products to be formed. And then the substrates are converted to products and released. And guess what? Because the enzyme can be reused over and over, the cycle continues. There's some conditions that affect enzyme activity, the ability of them to catalyze a reaction. For instance, pH and temperature. Remember, enzymes are proteins, and proteins are affected by pH and temperature. They're bonds. And so it turns out pH swings that are too high or too low can change the enzyme shape and ruin the activity, the catalytic activity. In general, if you increase the temperature not too high, turns out the enzyme and the substrate start colliding more often, and that actually increases the rate. However, you can't go too high or low pH or temperature, because then that is what we call denaturing, or stopping the function of that enzyme. It ruins the protein. Okay, so in general, notice that there is if we're talking about temperature here, here's the rate. Notice that the rates are specifically at certain temperatures that they are the best. So that really should be at the top there. But so this human enzyme, for example, operates best around this temperature, whereas this one in a bacteria operates quite higher. Okay. All right. So there's also pH conditions, and there's also an optimal pH. There's a enzyme called pepsin in our stomach acid, and notice it is optimal use in 2 to 3 pH, very low, and acidic. And then the basic condition, there is an enzyme called trypsin, which is in the intestine. Okay, so if you move trypsin into the place of pepsin and vice versa, they would cease to function. Pepsin works around 2 to 3, and if you put it any higher than that, it's going to stop working. There's also things called cofactors. Cofactors are non-proteins, so anything but proteins that bind to enzymes and help them. It's kind of like a family member helping me grade papers. It's a different thing and it's a different person, but we're doing the same job together and they're supporting me. Okay, and so some examples are zinc, iron, and copper ions. Okay, that's why we need it in our food and supplements. And there's also things called coenzymes. And coenzymes are cofactors like zinc, iron, and copper, but they're organic. They're carbon-based, and many vitamins are coenzymes like that. Okay, enzymes can also be inhibited competitively and non-competitively. Normally, we have a substrate matching up with an enzyme, fitting into the active site, and then the enzyme induces a fit. However, in competitive inhibition, there is an inhibitor that fits directly into the active site, and it inhibits by competing for the active site. It has more of a strength of bonding to the active site than the actual substrate does. And so it sort of is a bully and pushes the substrate out of the way. We also have non-competitive inhibition. This is an inhibitor that binds to a site other than the active site, but it still affects the binding. So notice this right here, this inhibitor can bind to this enzyme at this site, which is not the active site, and it can change the way that the enzyme shape is, and then it affects its function. Renders it not working. The concentration of enzymes in the substrates themselves can affect enzyme efficiency, not just changing pH and temperature. So if we increase the substrate concentration, so we would expect more of them to collide with enzymes, and we should expect more products. So typically it would just be this sort of black line here. And if we change the substrate and increased the amount of substrate, we should expect more product. Turns out an enzyme is not quite the same, because enzymes are the things that do the catalyzing. And so if you increase them, you're not going to increase the actual amount of product. We're only going to increase the amount of product starting, and then it levels off, because eventually there's a point where they level off. Okay, enzymes are also regulated to control metabolism. There's allosteric regulation. This is when a regulator binds to a specific binding site that is not the active site. So if we have an inhibitor binding to this enzyme at this point, this could render it useless, or it can permanently or non-permanently deactivate it. Sometimes they can also sometimes they can also activate it. Feedback inhibition is a type of allosteric inhibition where the end product turns off the enzyme. So after the series of steps, this one enzyme catalyzes a step, and then other steps keep going. Eventually at the end we come back and it tells the enzyme we need to turn off because we've made enough end product. Okay, in general, an organism's metabolism transforms matter and energy. Metabolism is a sum of chemical reactions in a cell. Living systems, like cells, require energy and macromolecules. Catabolism is a breakdown of the molecules into simpler ones. This process releases energy. It's usually the reactants at the top and then the products at the bottom, lower energy. Cats, I tend to think of cats just destroying everything, and so we can think of cats breaking down things. Anabolism takes energy, but it builds complex molecules. This is why they're called anabolic steroids that some athletes take to build muscle. Okay, and in general, energy is the ability to do work. It's the ability to move or cause change. And without it, we know our general appliances in life that aren't living, like lights and computers, they would stop working. It turns out living things depend on energy as well. Cells are moving, muscles are contracting, membranes are transporting substances. So we need that chemical energy from food molecules, and those are released by breaking chemical bonds. Energy cannot be created or destroyed, only transferred, and it cannot be reused. We need a constant source of energy. That's why we have to eat all the time. Free energy change. In general, free energy just means all the energy that's available or freed up to do work. Okay. Exergonic means that it releases energy, it's negative. Endergonic means that it requires energy, it's positive, delta G. Okay. And the important thing is just realize exergonic reactions, those are the ones that release energy. So products are on the bottom, reactants are on the top. ATP can power cellular work by taking exergonic reactions and using them to power endergonic reactions that require energy. For instance, ATP is strongly exergonic when it reacts with water. It loses a phosphate, it breaks off, and that creates a lot of energy. It releases energy. And this energy can drive another reaction. Okay, and that's what we call the hydrolysis of ATP to ADP. That's called coupling. Here's the structure of ATP. If we break one of those high energy phosphate bonds, notice that releases energy, and that can be used to do a reaction that requires energy. And so therefore we can kind of think like ATP is a charged battery. It loses a phosphate and it uses it up, but you can recharge the battery by adding a phosphate group again to recharge it. Let's talk about cellular respiration. The whole point is catabolic pathways yield energy by oxidizing organic fuels. What that actually means is that we are going to take a fuel like glucose, sugar, and we're going to take electrons off of it, and we'll use those electrons to power ATP formation, which is energy. Basically, we're charging the battery. Okay, this is usually done in a series of steps. They're regulated and controlled, so it's not random and there's no chaos. And there's really two main ways we can go about this. Fermentation produces ATP energy without oxygen. And if oxygen is present, we have aerobic respiration. I think of it's either aerobic, oxygen, air. And we usually call this cellular respiration, even though it's aerobic. We just put it together. The primary fuel for cellular respiration is not lipids or proteins, but actually sugar, glucose, okay, which is C6H12O6. It's going to react with oxygen that we breathe in, and then our cells create CO2 and H2O. Okay. Exergonic energy release from glucose adds a phosphate group to ADP. That's like charging that battery, remember? So we're going to use energy by breaking the ATP, and that's going to charge the battery. Okay. And so the whole point is we're forming ATP. Glucose, in order to form ATP, glucose has to lose electrons to CO2, and then the oxygen gains electrons is reduced to water. What do we do with those electrons that are taken from glucose? We're actually going to take them and put them into an electron carrier. This one is NAD+. It accepts 2 electrons and an H+ to form NADH, and then it is carried away to the mitochondria for later energy usage. Okay, let's do an overview of the cellular respiration process. Okay, so let's identify some locations. First of all, you should know this is the mitochondria. Okay, technically mitochondria in mitochondrion, but That's fine. Okay, and outside is the cytosol. Okay. All right. The first part of the process is in the cytosol, and it does not use oxygen. It's called glycolysis. Glycolysis literally means splitting sugars. Glyco meaning sugar, lysis meaning to split or break. Notice glucose is broken into two, so one glucose molecule is broken into two what we call pyruvate molecules. Glucose is a 6-carbon sugar. Okay, and we're going to break it into two 3-carbon sugars. That's what we're splitting it into. This actually generates a little bit of ATP by what we call substrate level phosphorylation. That just means enzymes are directly transferring phosphate groups. The pyruvate then has to go into the mitochondria and what we call pyruvate oxidation. Okay, by the way, noticing glycolysis, NADH is also produced, and this is going to travel all the way to a later endpoint in the mitochondria. Okay, and so pyruvate oxidation takes the pyruvate and turns it into something called acetyl-CoA. Okay, that process generates CO2. Okay, and it also generates some NADH, which carries electrons. So essentially, electrons are being stripped by NADH, and now we're going to transfer them to a later part in the cell. So this is happening at multiple stages here. So it's also happening in pyruvate oxidation. As pyruvate is oxidized to acetyl-CoA, that takes electrons, and those are transferred to the later process. Then those two acetyl-CoAs are going to be shuttled into something we call the citric acid cycle. It's also called the Krebs cycle. The citric acid cycle can also generate two ATPs by substrate level phosphorylation, enzymes phosphorylating ADP into ATP. But it also generates, most importantly, more NADH and also a new one called FADH2. It's also an electron carrier, just like NADH. The final culmination of all these steps ends in something that we call the electron transport chain in chemiosmosis, which is also mostly called oxidative phosphorylation. So oxidative phosphorylation uses a H+ gradient to pump ATP synthase, which turns ADP into ATP. That produces about the most ATP, about 26 to 28. Okay, so it depends on what shuttle. But if you add them all together, 2 from the glycolysis, 2 from citric acid cycle, and about 26 to 28 from oxidative phosphorylation, get about 30 to 32 ATP. Let's look at glycolysis specifically. Glycolysis takes the 6-carbon sugar, and we need to put energy into the system. We need to invest it. It's like how you need money to get a business started. You got to invest money to make money sometimes. ATP is going to power glucose and make it more reactive. And remember, as we take the triphosphate and use it up, the battery is now dead, and it's ADP. That's our investment, and we get an intermediate here, okay? All right, here comes the payoff phase. This is when we take 4 ADPs, and we're going to charge that battery for the cell to make energy in the form of ATP. That's called the payoff phase. Why is it called the payoff phase? Remember, we just produced 4 ATPs. Originally, we used 2, so that's a net of 2 ATPs produced. Okay. All right. Another thing to consider is the making. Where do the end products go? So first of all, ATP is used by the cell as energy, but at the end, glucose, that 6-carbon sugar, is split into 2 3-carbon sugars. It's split right in half. We call these pyruvates. So that's one pyruvate. That's the second one. These will work their way to the citric acid cycle. We also have NAD, which is able to strip electrons off of the intermediates broken down in glycolysis, glucose, and turn that into a charged form, NADH. NADH carries those electrons to the electron transport chain. By the way, exclusion statement from AP. In general, memorizing the steps in glycolysis and the Krebs cycle, the structures of the molecules, and the names of the enzymes involved is be all in the scope. Not necessary. Okay, after pyruvate is oxidized, the citric acid cycle, I'm going to call that the CAC, completes the energy yielding process of organic molecules. What that means is after pyruvate, so we produce pyruvate from glycolysis. Now it needs to enter into the mitochondria. Okay, so pyruvate enters the mitochondria from the cytosol, and there's a transport protein that does this. And then it's oxidized to something called acetyl-CoA. So we're going to take pyruvate and we're going to turn it into acetyl-CoA. Okay? This process loses a CO2. I'm talking about pyruvate oxidation. Pyruvate, as it enters, it will lose a carbon in the form of CO2, and it also gains this sort of COA part, and it makes this acetyl-CoA. Okay, so pyruvate is oxidized to acetyl-CoA. Notice that also generates us an NADH in that process. The important part is now acetyl-CoA is able to be funneled into the CAC, the Krebs cycle, citric acid cycle. What you need to understand is each turn of the CAC produces 2 carbon dioxide molecules. That's what we breathe out as humans. 3 NADH molecules, 2 here and 1 over here. 1 FADH2 that holds electrons as well. And it also produces 1 single lonely ATP. Remember we said earlier that it actually, citric acid cycle produces 2 ATP. Why is this? Remember, 1 glucose molecule makes 2 pyruvates. It splits into 2. And so 2 pyruvates become 2 acetyl-CoA. This is only 1 turn of the CAC using acetyl-CoA, 1 of them. Glucose breakdown makes 2 acetyl-CoAs. So we're going to multiply these products by 2. So really we make 4 of CO2, 2 of FADH2, 6 of NADH, and 2 ATPs. All right. So even though it's just 2 ATPs, again, energy is mostly in the form of the NADH, the electrons being carried to this process called oxidative phosphorylation. During oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. So basically, just like how water flows down a hill, so we have water flowing down a hill. In a dam, we have little turbines, remember? So we have turbines that spin when the water falls down. Turns out on the cellular level, that's actually how this process works. Instead of water, it's electrons. Electrons that come from NADH and FADH2. Those electrons are powering the movement of H plus that will spin a turbine. All right, let's label some diagrams here. First of all, we have these things called protein complexes in this cell. So this is a complex. Technically, these are as follows. They're 1, that's protein complex 1. That's 2. This is 3. This is, or actually, I apologize. One of them is missing. So I think it just goes 3 and 4. This is a simplified diagram. Okay. And so first of all, electrons are going to be transported from NADH. So remember, those come from the earlier processes. Glycolysis, citric acid cycle. It donates electrons and it becomes NAD plus again. So the battery is all used up. Okay, the electrons are transferred into these protein complexes. As the electrons move through the protein complex, we are going to pump hydrogen ions. Okay. So as the electron moves through the protein complex, we're going to pump hydrogen ions across into this space here. Specifically, we call this the intermembrane space. Remember, this is all happening in the mitochondria. Okay. Remember, this itself is the the inner membrane. Okay. This is what we call the matrix, the mitochondrial matrix. So we're going to pump all these H plus ions from the matrix into the intermembrane space. And the protein complexes are doing the pumping. And the protein complexes are powered by the electrons from NADH. Okay, so now we're going to start to build up a gradient. Okay, it's called an electrochemical gradient. So all these H plus ions are flowing into ATP synthase now. So as they sort of become way more concentrated at the top here, so we got to put a lot in. Okay, so we're putting all these H plus at the top in the intermembrane space. Now they need somewhere to go from high to low concentration. They flow into what we call this ATP synthase. Okay, so let's sum this up. This is actually called the electron transport chain. Electron transport chain, TC, I'll abbreviate. It pumps so the electron transport chain pumps these protons, and that creates a gradient across the membrane. Next step is called chemiosmosis. Okay, that's when ATP synthesis is powered by the flow of the H plus back across the membrane. So these H plus are going to flow down in there. Okay, and we're going to take our dead battery ADP, we're going to charge it and turn it into ATP, which is our energy source. Okay. These two processes together, all of them are called oxidative phosphorylation, electron transport chain, and chemiosmosis. Okay, let's answer some practice questions. Okay. So level 1 questions, these are basically comprehension questions that are not exactly AP sort of level, but it helps you understand the content. So let's look at this graph, and it says this graph most accurately depicts the energy changes that take place in which of the following types of reactions. The answer is D, it's exergonic. Okay, it's because the shape of the curve in the figure is going from the reactants on the top, higher energy, to the lower products. Okay, and therefore energy is given off. That's exergonic. Question number 2, enzymes are organic catalysts. How do they increase the rate of chemical reactions? Notice right here it lowers the activation energy, D. Okay, question 3 to 6. We use these terms once, more than once, or not at all. All these terms right here. Okay, step 3 describes a process by which an enzyme's function at the active site may be either activated or inhibited by the binding of a regulatory molecule at a separate site. If you remember, that is allosteric interactions. Allosteric interaction is when the enzyme is controlled at a site other than the active site. All right, let's look at 4. Molecule that is so like the normal substrate that it competes for the active site. Remember, that's called it's competition, competing. That's called competitive inhibitor, competitor. The process by which a metabolic pathway, number 5, is shut off by the product it produces. Okay, remember that is feedback inhibition. All right, let's look at 6. Molecule that binds the enzyme at a site other than the active site. Okay, 6 is technically non-competitive inhibition. Purpose of cellular respiration. Oh, the purpose of cellular respiration is in eukaryotes is to produce energy for cellular work in the form of ATP. Okay, so respiration is an aerobic process, meaning it requires oxygen. Notice A and B are wrong because respiration involves the breakdown, not the synthesis of carbohydrates, fats, and proteins. Okay, and D is wrong because oxygen is required, not produced. So therefore, the answer choice is 7 is C. Let's look at number 8. One glucose molecule provides enough carbons for 2 trips through the CAC. Using the figure on page 69 as a guide, how many molecules of ATP are directly produced in 2 trips? Okay, and the answer is going to be 2. Note that this question is framed per glucose, not per single turn of the CAC. Okay, so in the breakdown of glucose and citric acid cycle, 2 ATPs are produced. And in the course of the CAC, we produce 4 CO2, 2 ATP, 6 NADH, and 2 FADH2. Okay. Let's take a look at this free response question. I didn't cover photosynthesis directly, but we can still try it. An evergreen shrub, commonly known as a red tip phonetia, was used in the study to determine if there is a difference in photosynthetic rates from leaves exposed to direct sunlight versus leaves growing in the shade. It was hypothesized that the leaves exposed to direct sunlight would have higher rates of photosynthesis because photosynthesis requires sunlight than leaves in the shade. And the shade has less sunlight, obviously. All right, so to carry out this investigation, the same number of leaves were collected and placed in clear bottles connected to the oxygen sensor. Why might we want to do that? Remember, oxygen is a product of photosynthesis. So if you track how much oxygen we're making, that tells us the rate at which photosynthesis is occurring. Okay, so to maintain the same amount of light naturally exposed, collecting bottles remain in the area of the shrub where the leaves were collected to measure the rates of photosynthesis. And repeated trials were used with new leaves picked to the same region of the plant and with surface areas being the same. Okay, so look at the data. Notice the leaves in sunlight have a higher oxygen gas release. Look at graph 2. Concentration of plant pigments found in leaves. Notice that in general, the leaves in the shade tend to have a higher concentration of a certain pigment than others. Okay, look at this chart. This is the mean temperature in percent of the stomata openings between the leaves in the sunlight and leaves in the shade. Notice that leaves in the sunlight tend to have less stomata opened as not to let water out versus leaves in the shade. Let's describe the data that's used to measure the rate of photosynthesis. So graph 1 showed us that the rate is greater in leaves in the sunlight than in the shade, and that's because oxygen is released during the light reaction of photosynthesis. And that's when water is split and it is a waste product. Therefore, the amount of oxygen released represents how quickly water is being split, and it's like an indirect measurement of photosynthesis. Using graph 2, describe an overall pattern that is observed between exposed leaves to sunlight and those in the shade. The leaves exposed in the sunlight have a lower concentration of all of those 3 plant pigments, if you notice that. The student claimed that leaves exposed to sunlight would have a higher rate of photosynthesis than leaves in the shade. Identify one piece of data that seems to be contradictory. Provide reason. Well, graph 2 contradicts this claim. The leaves in the sunlight have a lower concentration of all 3 plant pigments. These pigments capture light energy from the sun, and so you would think there would be a direct relationship between their concentration and the rate at which photosynthesis occurs. But the chart 1 also contradicts this because it is shown that the leaves in the sunlight have a lower percentage of open stomata, and that would limit the uptake of CO2. And this would be expected to reduce the rate of photosynthesis. So that's why there's a little bit of a contradiction. Explain why having different plant pigments provides a means for plants to survive and reproduce in different environments. Okay, remember plant pigments like chlorophyll and carotenoids absorb photons for energy for photosystems. The photons are used to boost an electron to an orbital with greater potential energy and begin a transfer of energy to eventually make glucose. So different pigments absorb photons of light from different wavelengths. The idea is you want many rather than one because you can absorb as many different wavelengths as you possibly can. The more the merrier. Good luck.